Hello, everyone, and welcome to today's webinar, Break the Ceiling, Elevate Your Oncology NGS Testing with Expert-Curated Data. I'm Caitlin McCann-Jankovitz of QIAGEN Digital Insights, and I'll be the moderator for this event. Today's educational web seminar is brought to you by LabRoots and sponsored by QIAGEN Digital Insights. To learn more, visit digitalinsights.qiagen.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Q&A box located on the far left of your screen and click Send. We'll get to as many questions as we can during the live session, and any questions we do not get to will be addressed via the email address you provided at registration. I'd like to now welcome our speaker, Dr. Anna Krivokuca. Anna Krivokuca is a clinical laboratory geneticist with more than 10 years of experience in clinical genetics and cancer research. She has implemented innovative genomic technologies into routine molecular diagnostics in Serbia, and she's experienced in the interpretation of genomic data, variant annotation, and genetic counseling. Dr. Krivokuca is the founder and the head of the Department for Genetic Counseling at the Institute for Oncology and Radiology of Serbia. She completed her Ph.D. studies in genetics from the School of Biology, University of Belgrade, and her master's studies in bioethics from Clarkson University and Mount Sinai Hospital, Department of Bioethics in New York. Besides her everyday work in clinical laboratory genetics, she's been actively working on many research projects and has been mentoring master's and Ph.D. students. Her research is focused mostly on genetics and molecular biomarkers in breast and ovarian cancers. She has published more than 40 scientific papers during her career and has been awarded numerous research grants and awards. One more thing before we get started, our legal disclaimer. QIAGEN products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. For up-to-date licensing information and product-specific disclaimers, please visit www.qiagen.com. And with that, Dr. Krivokuca, I'll pass it over to you to get us started.
Hello, everyone. Thank you for having me with you today, and thank you for this introduction. I'm going to talk about COSMIC and HSMD for clinical NGS testing in oncology today. So here are my disclosures. And this is the webinar for today. I'll start off with the Precision Medicine Initiative, and I'll show you how it aims to enhance cancer treatment by using molecular profiling of tumors. So it enables personalized and more precision therapy based on specific genetic alterations of tumors. Then I'll continue with introducing COSMIC and showing a couple of examples on its use in clinical NGS testing. After this, I'll give you an overview of the HSMD, which still is not as famous as COSMIC, but hopefully I'll show you how this database can be really beneficial for clinical labs. And for the end, I'll show you how you can integrate both solutions in the unique pipeline for clinical variant interpretation. Okay, so let's start with Precision Medicine Initiative. And let me remind you of the traditional one-size-fits-all approach to treatment, where the same therapy was administered to all patients with similar disease characteristics. For instance, like it is shown in this scheme here, all, for example, colon cancer patients were given the same medication, regardless of the specific subtype of their illness. And now we know that this is not the best approach, since it led to varied response based on purely on statistics. And it resulted in serious side effects and even fatalities, while in some cases it proved to be ineffective. In contrast, the Precision Medicine Initiative represents a groundbreaking shift in how we treat and prevent diseases, especially in oncology. And it takes into account these individual differences in genetics, environment, and lifestyle. And a precision or personalized approach significantly advanced in fields like oncology. And we today tailor cancer therapies based on the genetic makeup of both the patient and the tumor. For this personalized approach, We use a wide range of tests, so we uncover the unique specificities of each tumor type, and we allow for the application of treatments and drugs that are specifically designed at least for a tumor subtype, if not for the individual patient. And of course, we couldn't do that if we didn't have a match between the technology development and science development at one point in time. So we still have NGS and massively parallel sequencing as the most important tool for precision or personalized medicine. Concretely today, I'm going to speak about NGS and molecular tumor profiling in molecular pathology, where we use this information for a lot of things, personalized therapy, biomarker discovery, pharmacogenomics, drug identification, drug effects, epigenetics, and so on. So basically, we screen a lot of tumors for a lot of information on their genomic profiles to actually tailor the treatment according to a specific subtype. However, even though testing for these somatic mutations in cancers is becoming a standard of care across many cancer types and almost everywhere in the world, there is a lot of challenges that still impede its broad implementation. And as you see here, these challenges include tumor heterogeneity, implementing drug combinations, clinical interpretation of coexisting mutations, clinical trial design, incidental findings, validation, and so on and so forth. And beyond these challenges specific to sequencing, analyzing a vast amount of genomic data, both in germline and somatic setting, and navigating the complex bioinformatics often lead to incorrect or incomplete variant classification. So we pose a lot of significant hurdle on clinical genomics or genetic scientists, and we must not allow for that because clinical genomics has high demands on data quality, reliable systems, and we need those for clinical laboratories. So we allow them to accurately identify potentially actionable variants for further patient management. And if we speak only about bioinformatics and analyzing the data, which we will today, it is an independent story. So we look into if we look for the information in knowledge bases that we have everywhere online. However, there have been a couple of studies in the last couple of years that investigated how these knowledge bases actually work and whether they share common data or they have unique information or what are their strengths or weaknesses. And all studies actually concluded that still there is no single knowledge base that fully encompasses all relevant data that we need. And these were studies from 2020 and on. And this is a daunting task, actually, for the variant scientists if they have to work and go through every database to look for the information, especially in the clinical setting. So we have a huge challenge to overcome. And for the molecular profiling of tumors, unlike looking for germline genetic information, pathogenicity is not something that we'll look for immediately. We look for the actionability status. So we have to assign the importance, the clinical importance to every variant that we detect. And we have to look through these databases that are available for this purpose. And sometimes I just want to remind you that pathogenicity here is important, but it's not always as important as actionability because sometimes even benign polymorphisms can be actionable or can be treatable with specific drugs. So what do? We have to classify every variant that we identify during NGS into one of 4 tiers in terms of their actionability. So tier 4, which is the green one, these are benign or likely benign variants actually, and these are not actionable, and there is no existing published evidence of cancer association between these variants, and these are not recommended to be included into the patient reports. Then on the other type of scale, we have tier 1 and tier 2, which are actionable variants. Tier 1, there are variants of strong clinical significance for which we have FDA-approved therapy that are included in professional guidelines, which is level A evidence, and we also have reliable studies from the experts in this field, which is level B evidence. So these are the most important variants that we detect. They have therapeutic, prognostic, and diagnostic value. Tier 2 are almost as important as tier 1. These are variants of potential clinical significance. We have FDA-approved therapies, but for different tumor types. And we have preclinical trials or few cases reports with level C and level D evidence. However, these are really important, and tier 1 and tier 2 should be considered clinically actionable. And the gray zone, of course, tier 3, these are variants of unknown clinical significance that are reported in the same or different cancer types with unknown clinical significance. And there is still no convincing published evidence of cancer association, so we usually do not know what to do with them. We actually have to perform regular follow-up. We have to re-evaluate VUS regularly to see whether there have been new evidence published about them and whether we can reclassify them either as actionable or non-actionable. So this can be a really important task and important work for clinical scientists to re-evaluate these periodically so we can issue new clinical reports. So we come to the story, our story for today. And this is the talk that is going to be surrounded on the use of 2 QIAGEN database. The first one is called COSMIC, which is short for Catalog of Somatic Mutation in Cancer. And the another one is HSMD, which is Human Somatic Mutation Database. Both are exclusively licensed by QIAGEN. And of course, we use them to support clinical NGS testing in oncology. And both of these databases are important, and I'm going to show you later on how, but we can use them to identify and annotate variants, to evaluate clinical significance, and we can use them as an aid for the challenges that I told you about earlier. First one is COSMIC, which is a comprehensive database of somatic mutations in human cancer. So it offers standardized data on somatic mutation. It gives you information about drug targets and biomarkers. It helps you identify driver genes and mutations. It helps you with clinical trials design, and it offers a lot of information that you can view easily, and you can export them, download them, and use them further on. Together with the COSMIC, I'm going to talk about Human Somatic Mutation Database, which is a web-based platform, which is also designed to help clinicians by using this extensive QIAGEN database. So it has an expert curation behind it, and this database includes more than 300,000 clinical oncology cases and over 1.3 million of somatic mutation. And they offer weekly updates on genes, diseases, variants, clinical trials, and drugs, all within a single integrated database. And I'm going to show you how you can work in each of these databases and how you can use them in your work. And at the end, I'm going to show you how you can integrate them both in your pipeline. So let's start with Cosmic. And before diving into examples of how you can use this database in your clinical labs, let me share some highlights from the latest release, which happened just recently in November 2024. This is version 101. And the previous one was in May. And I just wanted to make comparison between the latest and the previous release. So from May till November, they included 70,000 new genetic variants, over 6,000 new samples, so in less than a year. And this latest release, it focuses on somatic mutation data from whole genome and exome sequencing studies, and it also prioritizes rare cancer types and high need areas in oncology like pediatric cancers. They also focus on endocrine tumors and female-specific cancers. So the information that you have in Cosmic, it is integrated from sources like International Cancer Genome Consortium and clinical trials. So it's standardized. You can easy access to the data. You can download data sets. You can further use the exploration tools. And of course, what is the really important thing is that updates are made twice a year. So we are sure that we have the latest discoveries included in this database. And for those of you who haven't worked with Cosmic before, I wanted to show you how you can, through a couple of examples, how you can use this database in your pipeline. So you can, the first thing that you can do here is you can find clinically significant genetic variants that, for example, you look for. So this is the screenshot of the first page in this database. And there is a search bar where you can either enter whatever you need, or you can, and then you can go from that, or you can use the upper bar for search. Let me show you. So here is the search bar that you can enter, for example, the mutation that you're interested in, or you can browse the upper bar for the information that you want to see. For example, if you click on projects, if you click on the upper bar, it gives you the project information on the projects included in Cosmic, such as Cosmic 3D or Cancer Mutation Census. You can also go into the data to download the information that you need and look for the data sets that you need as well. And for example, here you can see actionability information, gene curation, and so on. And also you can go into the tools section where you can see cancer browser, genome browser, or information that might be of interest. So through the basic example, I show you how you can use on the database, for example, to prioritize clinically significant genetic variants. So you know that we all see a bunch of variations, especially when you perform mutational profiling of tumors. So this database will help you to highlight those that have major clinical relevance. So it's important to distinguish these key driver mutations from less impactful ones. You can also explore drugs, targets, gene expression, copy number variations, CNVs, fusions, but I'll show you that through the examples. If you work on ovarian cancer and want to find, for example, specific mutation, or you want to investigate the number and the types of mutations that are most prevalent in this disease type, you can use either search bar that I show you on the first screen, or you can use cancer browser through the tools part of the upper bar to enter specific disease data. Here I was looking for mutations in ovarian adenocarcinoma, although you can type in and search for any cancer type that you work with. And you set everything up in terms of selections, in terms of tissue, subtissue, histology, subhistology section. And you just click on go to get the data that you need. And first thing that you see in this section of the cancer browser page, you see a list of genes with statistics chart that shows the top 20 mutated genes for the selected tissue. For example, here I see a list of the most mutated genes in ovarian cancer, and the mutation frequencies are shown next to the gene name. So you have the list of the gene names and you have the list of mutation frequencies. The chart is ordered by the number of mutated samples examined. By default, it is drawn using known cancer gene listed in the cancer gene census. However, this graph can be changed to show all genes in COSMIC or only known cancer census genes by clicking the link under the graph. So here, and also the graph can also be redrawn to display only samples with mutations or only analyze samples by clicking here these blue or red dots in the bottom of the graph. So here you can use this feature to prioritize the clinically significant genetic variant that you found. So it highlights mutations with major clinical relevance. So immediately it helps you to distinguish key driver mutations from these that are not as important. So for example, in ovarian cancer, you should look for the information about these top mutated. Then you have another section, which is a distribution section, that displays a couple of charts again that show the distribution of different types of mutations for the selected disease. So you can mouse over the pie chart or table columns to highlight region of interest. For example, I wanted to see missense substitutions here and get more information of them. To get more information about these mutation types, you can click on them here. These are active links, so you can explore them further. And have much more information, additional information on the relevant samples and mutations. So I saw substitutions here, and you can explore drug targets, you can explore gene expression, CNVs, and you can understand each mutation's role. You can also see the types of mutations here, and you can explore, let's say, drug targets. You can look into a table of mutations found for selected disease. This table also provides the ability to search, sort, and export the table in, for example, CSV format. So here you see, in the first column, you see gene names. You're seeing gene names in which the mutation has been found, and it links you directly to gene analysis page for more details on that gene if you want to look further into. Then you have a samples and the sample name in which the mutation has been found. Again, active link. In the third column, you have the name of the mutation according to AGVS. And the description of each type can be found on the following link, of course, if you click on the active link here. And in the last column here, you of course see the information of the impact of that particular mutation on the protein. This table shows the sample name and mutation count for the tissue or the disease selected. So you can see how many mutations have been detected in particular samples, which can be important in terms of statistics and epidemiology. And I wanted to highlight a specific feature of COSMIC, which is COSMIC actionability. It is a standalone product, but I think it's really important for clinical labs because it provides insights into the genes, drugs, trials, mutations. It links mutations, diseases, and drugs. It identifies existing and emerging therapies, so it's important for clinicians. And of course, again, it annotates, it gives you the information about annotation and actionability of those with clinical relevance. and it highlights these cancer type specific drug responses. So you can see whether you can use it for application of specific treatments. So it helps clinicians to prioritize these information and these variations that can be important for their patients. And the latest release from November, it included more than 500 curated genes. So you can go into the gene table and you can click on the gene that you want to see, and you can look for a mutation that is actionable, which is linked to information about drug sensitivity, resistance, or whether there have been treatment that are FDA approved or not. So I think this is the really important feature of for clinicians. And what is really, really important, I think that COSMIC simplifies mutation reporting because it gives each mutation a unique ID. So it's easier for both researchers and clinicians to communicate between them clearly. So in the sea of so many clinically important genetic variants, we have IDs that help avoid confusion, and they ensure accurate data reporting and provide quick access to the details like genomic location, clinical significance, and so on. And you can use this information to standardize your clinical reporting across studies or with the patients. You can use to promote collaboration, and you can use it to integrate information with other genomic databases. So I think it's really important because I've seen a lot of different uses of IDs for these variants, so it's difficult to sometimes to communicate about them. So I think this is something that really helps and also supports better treatment decisions. This is actually sort of a case study or a story that I wanted to share with you. Because the latest version highlighted the information about radiation and the occurrence of papillary thyroid cancer, or PTC. I want to remind you about 1986 catastrophic explosion that happened in Chernobyl in Ukraine. And this was the accident that exposed millions of people in Ukraine, Belarus, and Russian Federation to these radioactive contaminants. And a lot of lighter particles also were carried by wind across these countries and also parts of Scandinavia and Europe. And approximately more than 350,000 people were evacuated due to this disaster. And these efforts are still there to resettle the affected areas. They are still ongoing today. So I wanted to show you how COSMIC included the important information from the study related to this accident into their database and how you can use it. So this study that was published in 2021 in Science was actually groundbreaking. So this was the first study to comprehensively analyze the genomic landscape of PTC that is caused by radiation exposure from the Chernobyl disaster. And it provided the unique insights into radiation-specific DNA, double-strand break signatures, and molecular mechanisms behind it. It analyzed 383 samples from individuals exposed as children to ionizing radiation from this disaster. Here, I wanted to show you a couple of important highlights of this paper. This is the landscape of somatic alterations in PTC that are caused by radiation dose from 131 iodine exposure. And they detected more than 300,000 of somatic mutations. They divided them into coding and non-coding variants. And BRAF V600E mutation was the most frequent, with 45.2% of all detected mutations, and it was followed by mutations in the RAS family, NRAS, HRAS, and KRAS. Here you can see distribution of radiation dose from iodine exposure by driver type and pathway. and relationship between radiation dose from iodine exposure and selected genomic characteristics by age at exposure. And while most cases were driven by mutations, 59%, others were driven by fusions, which were more prevalent in individuals that are exposed to higher radiation levels or diagnosed at younger age. So this is something that was really important as a conclusion. Also, they saw pronounced and showed pronounced evidence of radiation-related damage that was observed for individuals that are exposed at younger ages. It was consistent with epidemiological analyses that have identified higher thyroid cancer risk with radiation exposure at younger ages. And regarding drivers, most tumors had evidence for only a single non-oncogenic driver, which involved the MAPK pathway in nearly all cases, which is also consistent with previously published studies, but of sporadic PTCs. They have some really interesting conclusions here. And these results have important implications for radiation protection and public health as well, particularly for low-dose exposure from two perspectives. First of all, they highlighted the challenge that still exists to identify a reliable biomarker for distinguishing radiation-induced tumors from those that are caused by other factors. And their findings support something that was known, such as linear dose-response relationship for the key molecular features associated with radiation exposure. So I suggest for you to take a closer look at this paper if you are interested, of course. But here I wanted to see how Cosmic integrated the information and these comprehensive results that was published here. So what did I do? I looked for supplementary material, and I looked at something that was of interest to me. I looked into the distribution of these rare, potentially protein-damaging variants that are found in selected genes in the study population. And this is the table from the supplementary material that you can see a lot of information about the gene, about the mutation, about the type of mutation, and what was important about the reference population data from GNOMED or the study population itself. So I wanted to see whether I can find more information about one particular gene, GTF2H2, that is involved in basal transcription and nucleotide excision repair in the Cosmic database, which is a gene with the rare mutations not reported in population databases, but reported in this particular study. And of course, I went directly into the Cosmic database and I used the search bar to look for GTF2H2 gene, and I had the gene view overview information about this gene, which I expected to see. However, I wanted to see the tissue distribution and mutation distribution in different tissues of these particular genes, to see whether PTCs are one of them, so you can explore the distribution in different tissues. And I wanted to see, of course, mutation distribution, to look at the ones that were detected in this particular study and to further investigate the types of mutations, which was all there, of course. Here, as I showed you before, I wanted to see the type of variants in this gene. I wanted to find the mutations that were detected. I wanted to see which mutation type it is, and I wanted to actually further look into these mutations. And when I looked at the references, one of the references is the study from 2021, which is the study that I showed you before. So the thing is, I showed you that you can use Cosmic for your clinical labs to annotate, to look for the information, but you can also be sure that the information that you look for is updated and that you have the latest information on your variants. Besides COSMIC, there is another database from QIAGEN that is really useful for clinical setting, and it is called Human Somatic Mutation Database. And this is the first screen of this mutation, and it's a comprehensive resource for clinical labs because it provides weekly updates. So you have the information on genes, on different alterations, on diseases, on drugs, on clinical trials, or within a single integrated database. So you can look for clinically relevant mutations in genes, and you can synthesize findings from drug labels, trials, guidelines, and so on and so forth, but I'll show you a couple of examples. The first thing I use HSMD for is to annotate variants and to look for these that are clinically actionable. If you do it manually, this is the example of one BRCA1 missense mutation, and you can look into the gene, for example, and look into the alteration type if you want to investigate the types of alterations in this gene. You can look for those that have specific functional impact, or you can directly search for tier 1 or tier 2 mutations detected in BRCA1. What you can do, you can just check out the actionability distribution, and you can see how many tier 1 or tier 2 mutations are detected or included in the HSMD in this known gene. Interestingly, most of the mutations in BRCA1 are actionable tier 1. So what else you can see? You can find your mutation of interest. You can look into the alteration details. You can look into the references, and you can gather the evidence to be sure that it is the actionable variant that you can use for clinical reporting. How does it look like? So here you can see the alteration details. You have direct external links to other databases regarding biochemical impact. There is information on different in silico tools. Also, which is I think most useful for clinicians and clinical labs, you can look into the drugs and clinical trials that are related to this particular mutation. For example, I wanted to see for which drugs this mutation is sensitive for, and I wanted to see ESMO guidelines that recommend therapies for this mutation. To go further into the details, I was looking for breast cancer clinical trials, and there is the list of clinical trials, different phases. You can see the information on the drug that is used. You can see the information of the genetic biomarker, and you have ID of clinical trial. You can click on them. These are active links, so everything is in your hand. At the end, I classified this variation as tier 1 regarding actionability. I had all the alteration details. I had the functional impact. I had information about the ongoing clinical trials in breast cancer, which I was interested in. I also found drug associations and found source or references from QIAGEN knowledge base. So this is the first thing that you can use HSMD for, to manually annotate and classify variants and to directly link them to drugs and clinical trials. What else you can do? You can evaluate complex genomic reports. Sometimes you outsource your NGS. Sometimes you receive complex reports with a list of different variants. So it's difficult for clinical scientists to draw the clinically important information for clinicians. So this is the tool that you can use. You can use to investigate the clinical relevance of these alterations. You can find treatment options and mutation frequencies. So sometimes it's just easier to manually look through the information than to look to pay or find complex softwares for it. And also, many complex reports nowadays include the information about variants of unknown significance. So the important thing, as I said before, is to look periodically whether there is new information about these variants. So you can use HSMD for. So there is a list of variants that you have that you need to evaluate or reevaluate, and you can easily do that in HSMD. So this is the structured approach, actually, that facilitates the identification of actionable variants. So you integrate the HSMD content with the real-world data, and it actually helps you to interpret complex genetic reports. So overall, for example, I had a complex report. This was a lung cancer patient, and our clinicians wanted to see, and they actually asked for the information about FGFR2 mutation and JAK1 mutation because they thought they can use it in the patient management, and it was easy for me. So I found it really easy into HSMD. FGFR2 was not actionable, it was tier 3, so I put it in the database to look for more information later on in the future. However, JAK1, this was tier 2, clinically observed in endometrial and colon cancer, so some off-label therapies could be considered, and some of the clinical trials that I searched for the clinicians might be considered as well. Of course, another thing that you can do, you can use HSMD not just to annotate and evaluate particular variant, but you can look at the variants in a specific type of the disease. For example, if you have a lung cancer patient, so here you can just type in and look into the disease tab, lung cancer, and see what is the most prevalent here, and look for the mutation that you have, and of course look a particular mutation. But similarly, as in Cosmic, you can evaluate the types of the mutations and the frequencies in the specific cancer type. As I said before, I really think that VUS or variants of unknown significance that we see a lot are really important to evaluate periodically. So you can use HSMD for that. It's easy, you just type in the mutation, the variant, and you see the classification status, so updated one. And also, these complex reports usually they have secondary or incidental findings. So sometimes these reports might come back with information you haven't been searching for. So these secondary findings are common in the NGS era. So we can use HSMD to evaluate them the same as the VUS, and we might be able to maybe establish their new roles in the disease and maybe to reevaluate their therapeutic potential. So these are all really important for the clinical oncology. What else you can use HSMD for? You can use HSMD as sort of an app because you can quickly find information on tumor genomic profiles. You can determine variant actionability and match clinical trials with a specific genomic profile of your patient that you have in front of you. This is really important because it can be significant aid, for example, for mutation tumor boards and these ad hoc queries that you have. So you have information that you need the answer to right now, and you can get it using HSMD. And for these MTBs, I think the diseases, drugs, and trials tabs are the most important. It offers you the description, although maybe the disease is not as important as drugs and trials because you already have a patient with the disease that you know of. So you might want to look for a list of drugs, gene alterations, list of clinical trials, and the geographical position of the clinical trial studies, novel information, maybe off-label therapies, and you can search for clinical trials based on a phase, status, type, country, location. And for example, I just wanted to show you on which questions they're most often heard on the MTBs. You can get a quick answer to. Is this patient, my patient that I have in front of me, eligible for targeted treatment based on the molecular profile that he or she has? Is there a clinical trial that could be considered for the patient? Where is the clinical trial? And is there an off-label therapy that I can suggest or recommend or even try for my patient? So this was the story about two databases that you can use for similar purposes in clinical labs in oncology. I wanted to show you for the end how you can integrate both solutions and to make a pipeline which is reliable and that you can reliably use in your everyday work. So as you saw, both databases, they serve as valuable resources to enhance the effectiveness of somatic NGS testing workflows, and they allow you variant interpretation with mutation data and clinical insights, which is really, really important. So you can use COSMIC, for example, in the initial phase of complex bioinformatics analysis, because it plays a critical role in prioritizing clinically significant genetic variants. It also helps you to annotate actionable mutations. It evaluates somatic frequencies, and it can map genomic coordinates. So you can have a reliable data for use in reports. So this can be the first part of your pipeline, that you use COSMIC to generate reports after you have your sequencing done. After you have a patient report, then you can use HSMD, complementing COSMIC, of course, because HSMD can provide you with insights by extracting report relevant information. You can evaluate secondary findings that you have. You can identify off-label treatment options. You can locate region-specific clinical trials. Additionally, HSMD supports molecular tumor boards recommendations, so it can help you to guide personalized treatment strategies. So together, they can make a unique bioinformatics pipeline for a clinical lab. So after the sequencing, use COSMIC to generate report, then use HSMD to interpret these complex reports and to have therapeutic recommendations and even specific suggestions to your patient management. And with this, I would like to thank you for your attention, and hopefully I'll show you how you can use these databases and how they can be useful in your everyday work. Thank you once again for being here with us today.
Thank you, Dr. Krivokuca, for your informative presentation. We will now start the live Q&A portion of the webinar. To the audience, if you have a question you'd like to ask, please do so now. Simply type your question into the Q&A box located on the far left of your screen and click send. We'll answer as many of your questions as we have time for. All right, let's get started. Our first question is, can I use the actionability feature in COSMIC if I don't have the license?
Yes, thank you for the question. I think this is really important for everyone to know that you can all use actionability feature in COSMIC. It is a standalone product, as I said, but you can click on the actionability data from the official website. And in case you don't have an account and you are, of course, non-commercial user, you can just register and you should be able to download any actionability data. And if you're a commercial user, you already probably have your account via QIAGEN site. So I suggest everyone who doesn't have the account with the COSMIC to create one and to try and to check out the actionability data, because I think it's a great feature in this database. So yes, my answer is yes, you can download actionability data even if you do not have the license.
All right, thank you. Next question. These two databases seem similar. Do we need them both? And what do you suggest about their use?
Yeah, they seem similar. They seem similar to me as well. But when you dig deeper, you can actually see by yourself how you can use them both. You can also use them individually, but you can also integrate them in a unique pipeline that I showed you in the last couple of slides. As a sort of a tip, how would I? Of course, if you have the option to use them both, it's great. As I told you before, the cosmic you should use for the first part of the bioinformatics analysis. You can create reports, and then after you create report, then you can interpret these reports with HSMD. But as a sort of a tip, I think that Cosmic is excellent for identifying mutations, for understanding cancer biology, and informing experimental designs in the clinical, in the setting of clinical research. But if you work in the clinical lab, you can of course use it for the mutation identification, and then as a first part of bioinformatics process. If you need it for clinical use, of course, I would suggest to cross-reference it with other databases. In this case, HSMD, it's more than enough to just create a unique pipeline from the beginning, from the sequencing, from the mutation detection until the end, until you have a final report, clinical report. So first use Cosmic as a first part of the pipeline, and then you can use HSMD to interpret report and to link the mutation data with the actual patient and the clinical management of that particular patient that you have in front of you.
Okay, on to the next one. Can we upload VCF files into HSMD?
Probably someone from QIAGEN will correct me if I'm wrong, but I think no. This is not a software. This is a database. So you will need the software for VCF files and for generating complex reports like other QIAGEN products like UCI, for example. But HSMD is database, and it is a comprehensive database that gives you information about particular mutations. So you can type in the mutation and look for it. You can see the link between the mutation and the drug and the clinical trial, and you can, of course, search from the clinical trials down to the mutation and so on and so forth. So HSMD is not designed to give you the complex report on many mutations, such as extracting them from VCF files, but maybe sometimes in the future it can be available too, we'll see. But for now, just use it as I show you for particular mutations and anything that you want to link and connect with that mutation.
All right, thank you. Okay, next one. In your presentation, you said that no single should be used exclusively. How do we know we can trust either Cosmic or HSMD?
Yeah, everybody says that for a long time, that no single knowledge base should be used if you work in a clinical setting. And I do agree with that because I've tried so many and I've seen so many strengths and weaknesses. But here, of course, and sometimes you need to just work with what you have. Are they trustworthy? Yes, but I suggest to combine them both. I mean, Cosmic, you probably know about Cosmic for a long time. It is widely regarded as a trustworthy and reliable resource for cancer genomics, especially for research. Again, it is curated by the Wellcome Sanger Institute, again, highly respected research organization. And if you ask me about the strengths of Cosmic, there is curation and peer review. We have data from scientific literature, high throughput sequencing studies, cancer research projects, and everything is curated by experts. So we have high data quality and accuracy. Also, you have a comprehensive database with a lot of information and resource for identifying driver mutations and cancer-related genes. It is regularly updated. It is transparent, so you have everything is clearly described. You can access to the reliability of the data. It is again widely cited. So it's not something that it's not trustworthy. It's more of a question whether it's enough for what you do or you need something more. That's why if you work in the clinic, I would suggest to just add information from HSMD, which is again database from created by QIAGEN with a really great reputation. Again, it has curated and comprehensive data and so on and so forth. If you ask me about the limitations, of course there are limitations with every database, including Cosmic and HSMD. Cosmic is extensive. However, maybe especially for these less studied cancers or rare cancers, even though they've included rare cancers in this latest release, it may not capture every mutation or data from all cancer types. So sometimes maybe these understudied cancers are not as included in the database as others. Also, it relies heavily on published data and specific research projects. So again, it is going to introduce biases towards well-studied cancer or genes. But I think it's the same with every database nowadays. So again, these are trustworthy databases. You can use them individually or together. And depending on your needs, especially if you work in the clinical labs, you can combine them both and be sure that you have updated and reliable information.
Okay, and this next one is kind of related. How does Cosmic make sure that mutations are mapped to precise nucleotide positions with consistency?
I think it's not a brainer. I think they use standardized reference genomes such as GRCH 37 and 38. Also, these mutations are annotated with you have coordinates, so you'll see when you perform the search in the database. They cover both coding and non-coding regions of the genome. I think they also integrate genome browsers like Ensemble and UCSC, so you can actually visualize mutation locations. How else? I think it also enhances accuracy by cross-referencing referencing with other genomic databases. It's DBSNP is also available there. So there is a huge supporting system for the detailed research and clinical decision making in cancer genomics. So I believe that this is not something that you should be worried about. Again, just do a little bit of search and you'll see everything you need to know about the particular mutation.
Okay, great. And with that, it looks like we have time for just a couple more. These last two are both regarding HSMD. So first one, can HSMD be used for in-house bioinformatics pipelines?
I think it probably is something that HSMD is perfect for. Because if you have a small clinical lab, for example, and if you have your own pipeline, you know how burdensome for your men and your people working in the lab is to annotate genetic variations. So you can use HSMD for speed up this part of the process, and of course to be fast and to be reliable. So I think it can completely be part of the bioinformatics process, and it can of course use HSMD for the things that you performed manually before. So you probably searched for information about the variations following ACMG guidelines. It takes a lot of time. It takes a lot of manpower. Sometimes it takes a lot of people to analyze the data together. You can use HSMD for this. It has information on more than 1.3 million of mutations, if I remember correctly. It is expertly curated. it gives you a solid foundation for any type of the analysis of variant analysis. It's regularly updated. It has real-world data. It covers biological relevance, clinical associations, drug responses. It can help you prioritize actionable mutations, support secondary finding evaluations or VUS evaluations. So you can use it as a part of the pipeline in terms of just have it in your bioinformatics team. And when you have the data from the NGS, you can just do a search through the HSMD and give your people just time to interpret these reports and to give the feedback back to clinicians. So it can be really helpful as part of the bioinformatics process in-house if you create one.
All right. And our last question of the day, is there a free/trial version of HSMD?
I think they've been talking a lot about, and I was also interesting to know whether there is a free version or any sort of trial version of HSMD. If it's not out there yet, I think it soon will be. Of course, I think it's really important for anyone to try out the database to see whether it fits the needs. So please contact QIAGEN. I think that a sort of free version will be available soon if it's not yet. And you can just take a look of what it has to offer. And then if you think it's useful for your clinical lab, then you can then see what else is out there and you can negotiate for the full license.
Okay, awesome. And that will conclude our live session today. Thank you again so much, Dr. Krivokuca, for your time and your important work. I'd also like to thank the audience for joining us and for their participation in Q&A. Any questions we did not have time for today or any that are submitted during the on-demand period after will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues and any who may have missed today's live event. Until next time, stay safe and be well, everyone. Goodbye.
QIAGEN.
Sample to insight.